So, okay, so today is a question and answer on clinical trial. That's not for today, but I hope it will be for very soon in Myhre. So last month we talked about, oh my God, I removed that. Sorry. Can you still see my screen with the presentation or not?
Yes.
Yeah.
Yes, we can see.
Sorry, I did something wrong. I can see it. Okay, now I can see it. So last month we talked about how to measure potential treatments before humans. So in models that are based on cells, organ that we call organoids, and in vivo models. So when we have that first part that is tested outside of human, so before human usually, then if we have everything ready, we can go to humans. So what does it mean, everything ready? When can we start a clinical trial? When the FDA agrees that there is a good chance that the treatment can be tested in human, it's only when it is shown that it is safe and that it will have a probable chance of efficacy. So those are the two main blocks that the FDA wants to see. Is it going to be dangerous for the patients? Yes, no. Is it going to change patient lives and manifestations? Yes or no. So to prove them that, you need to have what we call preclinical data on the molecule or the treatment you want to test. So that's what we talked about last month, sorry. So you need to have data and proof that it works in cells and animal models. So you want to prove that the treatment is efficient to decrease the disease manifestations. It depends, for example, if you want to target tracheal stenosis, you will have a very specific clinical trial, and you want to prove that this treatment preclinically, so on cells and animals, is going to change tracheal stenosis. If you want to treat in a chronic way, Myhre syndrome in general, then it's several manifestations, and you need to have more proof that it's going to change the patient life in different type of issues that they have. So those are two different type of proof. And you need to prove also that it's safe. So it's what we call toxicity. So you can have that tested in cells and animals. So you are going to do what we call PK/PD in animals. So that's what we talked about last month. So you test the toxicity, and if you can prove that it's not toxic, then it's one more step towards human. And you need to prove also that you can produce that treatment in what we call a GMP conditions, which means that it's going to be prepared, the treatment, the molecule, or the whatever treatment, if you want to use cell therapy, gene therapy, you need to prove that you can produce it in conditions that can be applicable in humans. So it's sterile, it's extremely controlled, and it costs a lot of money to be in GMP conditions. When you go to animal and you test it in animals, you don't really care if it's in GMP conditions, so the production of the molecule is way less expensive. If you are looking at repurposing, you just need the evidence of efficacy because the safety has already been done before. So does everyone know what is repurposing? Yes.
Yeah.
Okay.
Yes.
Okay, so all the testing has been done already.
I do. Maybe not everybody does.
Because I don't see you because on mods I have only one screen. That's the issue. Well, scream if you don't if you don't know. So the repurposing, the phase 1 and usually phase 2 has been done, so we know that it's safe not only preclinically, but it's safe also in humans. So it saves a lot of time in that aspect. And then you need also to prove to the FDA that you have a clinical trial design, that everything is ready, the protocol is ready, you know how and what to measure. So when you have all those data, then you can go to the FDA, file what we call an IND, or file file to get the authorization from the FDA or the EMA in Europe to run a clinical trial on human. I go to the next. Okay. So how do we measure this safety and efficacy, and how do we get to the clinical trial? First, the FDA, to take their decision, they like to know the patient's voice, as they say. So that's what we call the patient listening session. So you go to the FDA and you share the patient experience and priorities. Ines, we did that at the foundation, and Ines participated in the patient listening session together with 7 other people. Am I right, Ines? I don't remember how many.
Yeah, there was about 6 or 7. Yeah, there's about 6 or 7 of us. Yes.
So we've done that so that the FDA knows the Myhre syndrome, and they know exactly, they know from different perspective and different type of patients how it is to live with Myhre syndrome. So that's one point. And then we need to know about the disease. So what we call the longitudinal natural history. And that's studying patients, taking all the measurements year after year, following the same patients to understand, in fact, what's the progression of the disease without treatment, of course, and understands, really characterize the patient. What do they have? Which kind of manifestation? Is it progressing fast, not fast? What's the difference between pediatric and adults? So that's the whole history, the natural history of the disease. At the same time, now more and more, we want also to collect what we call real world data. And that's mainly in rare disease. That's coming from all the neuromuscular diseases, because they realize that it's not only going to the clinic and having some very specific clinical test that you can understand the disease. You need to follow people during their life and understand the small characteristics of their disease in their everyday's life. How do they go down the stairs? How do they wake up in the morning? All this kind of, how can they get into their car? All these kind of everyday's life events that are characterizing the life of the patient. That's also extremely important, and that's something now that we can follow, and we can compare a treatment without the treatment on the real world data. That's very important, because then we realize that it's in those small differences that we can measure the efficiency of a treatment, rather than a 6-minute walk test, for example, that will not really reveal the small impairments that you can have in your life. So we will not be able to measure the important things, because the important thing for the patients is the real life. It's not a 6-minute walk in a corridor of a hospital. So that's the things that we really need to understand. Then we need to define the population. As I mentioned before, you have the pediatric, you have the adults, you have some population. I mean, when I say population, it's the different groups in a disease. Look if they are all the same, they follow all the same progression, or the different type of progression you can have. In Myhre, you can have progression in heart, in respiratory, or not, just remain with joint issues or intellectual developments. So you really have to define where the treatment is going to be tested in what kind of patients, what's the population. Then you need to choose the type of clinical trial design. So you define the exclusion inclusion criteria, who is going to participate, for how long, what are going to be the control? Do you compare patient with the treatment with patient that have a placebo? Do you compare to the patient themselves in the past when you measure without the treatment and then you compare with the treatment? You have different arms, so you can have different either manifestation that you look at during the treatment or different type of patients. So when you have chosen the clinical trial design, you have also to select or create, and that's what we probably are going to do at the foundation, create measures that are relevant to the disease and the patients. And the way you measure things, that's called endpoints, and that's what are going to decide if the treatment is efficient or not. Do you have any question on all these different points? No?
I don't. This is very interesting. I do not, but maybe somebody else does. This is very interesting.
Just shout because I don't see you. I'm sharing just my screen. Okay, so I'll go on. So what are the clinical trials for rare disease are different from the larger disease population like diabetes, for example, or other type of disease where you have a lot of patients. In rare disease, rare is the challenge. And what we see is not only the limited patient population because it's a rare disease, it's also that most of the rare disease are very heterogeneous, which means that you have one patient that will have cardiac issues and the other one will have, I don't know, depression. So that makes that is a real challenge because you already have a limited patient population, but then you even decrease the number of patients if you want to go for each different manifestation. So the heterogeneity is a real issue in that sense that it decreases the number of patients where you can test the compound or the treatment. And at the same time, there is this heterogeneity because it's a multi-systemic, so it's going to act on different parts of the body, like cardiac, respiratory, immune system, neurodevelopment, and so on and so on. So what are you going to look at? It's multi-systemic. You don't know which one you want to choose. If you want to treat the chronic, the whole syndrome. So that's also a challenge. Limited patient population, I'll talk about that also. And the next challenge is chronic versus acute. So it's not only Myhre syndrome, it's not that you have one problem. You have different problem chronically progressing along your life, and you have acute problem. Like tracheal stenosis, for example, is an acute problem and you need to act now. So the way you are going to choose the treatment for tracheal stenosis or an acute problem or pericarditis or this kind of acute life-threatening issues, the way you are going to choose the treatment and the way you are going to measure and the safety issues and everything in the design of the trial is going to be different from the chronic treatments where you want to decrease fibrosis. You are going to take a treatment for years and years versus when you have an acute issue, you are going to take the treatment maybe for 3 months until the issue is resolved. But the chronic, it's for years and years. So the safety is different. What you are going to measure is different. And the population is going to be different. It's larger than acute issues. I'm going to change slides, so you tell me if you have any questions. No. So the type of clinical trial design that we have in rare disease are what we call non-conventional. So there you have different aspects that in Myhre we are going to combine, in fact, and depending on the type of treatment we want to choose, we are going to take a little bit of everything. So you have what we call adaptive trials. These trials allow for pre-planned adjustments, sorry, based on interim results. What does it? So that's a sentence from the FDA. So the FDA, when you apply, wants to know exactly what we call the protocol, so the way you are going to do things. And you cannot move from that. Otherwise, they will just cut you out. So you need to follow exactly what is planned. But sometimes in rare disease, you want to adjust and you want to modify. So you can do that. You're allowed to do that if you have planned that maybe you are going to change things according to the results that you have after 6 months, a year, or a few months. You can look at the results and say, okay, this is not working at that dosage. I'm going to change the dosage. This is not working, just one treatment. I'm going to add another treatment on top of it. You can imagine a lot of different adjustments that you can do, but you still have to plan them in advance and propose them to the FDA. So that's a way to be more efficient for rare disease. That's the liberty we have to adapt the trials. The basket trial is also another one that we can have in Myhre, because Myhre, being smart for gain of function, you touch the whole TGF beta world and family, meaning that there are other diseases that could benefit from the same treatments than Myhre syndrome. So the basket trials is in fact when you treat different population with the same, so different diseases or manifestations. Even in the same disease, it can be different type of manifestation with the same molecule. So you start from one molecule and you add, it's a basket because you add different type of manifestation or different type of disease patient. It has been done up to now in tumors, in rare tumors, where they have a large population that can have different type of tumors, and they go, in fact, with the same drug, they look at one tumor, and then when they see that it doesn't act on that, or even if they see that it acts, they can go to another tumor and another tumor and another tumor. For us, we have fibrosis, and we can adapt what they do in oncology on multiple types of rare tumor, we can do on multiple types of fibrosis. So you can have liver fibrosis or muscle fibrosis or tracheal stenosis or different type of fibrosis with the same molecule, which means that in that clinical trial, you will have enough patients, enough a variety of patients, and you increase the number of patients. So first you get way more interesting from the pharma company than when you are only 10 people. And at the same time, they can really do large statistics and have a better chance to see if a treatment is efficient or not. So that's a good way. There is another type of trials which is called the crossover trial, where the participants receive a sequence of different treatment, and they act as their own control. So the patient is its own control. So you start usually with no treatment, then you take a treatment, then you stop. You wait 6 months, you see the difference, and then you can start with another treatment. So that's a way also to deal with the fact that you have a limited number of patients. With this kind of trials, you don't need to punish, I would say, a part of the patients on being on the placebo. Then you can give the treatment to all the patients and not have half of the patients that are on placebo. So that's huge for us. It's the other one. Did you have any question for the one before? Maybe I can.
Are we leaning towards one trial over another?
We are going to, so that's where I'm going on the next slide. We are
Okay.
going to take a little bit of everything, and we are going to go. So for each treatment, you need to file a different file to the FDA. Every
Okay.
time you have to do it again for every single molecule. And every time you can adapt because you have chronic versus acute. And also, if you are doing gene editing, it will not be the same clinical trial that if you are doing a small molecule.
So
So every time we are going,
you can use more than one trial.
oh yeah, yeah, yeah. Every time it's a little bit different, and we are going to have a protocol that takes a little bit of adaptive, hoping that we'll get a basket trial with other diseases that are concerned with fibrosis, and we'll take a little bit of crossover and another one that I'm going to talk about now
Okay.
for Myhre syndrome. So the thing I want you to understand is that a clinical trial is very dynamic, and every time it's different because every time the manifestation, the treatments, everything is different, and what you want to measure maybe it is different. So it's not something that you decide on in 2025 and you will have the same protocol in 2028. It's very dynamic. So for Myhre syndrome, I was saying before that you have two really different type of clinical trial that you want to think about. It's the chronic treatment versus the acute treatment. Let's talk first about the chronic treatments. What we want to do in Myhre is a composite domain clinical trial. So we are not going for a specific focus. It's really broad. And we want to evaluate the treatments and efficacy of the treatment across a set of distinct outcome domains. So what does it mean, outcome domain? It's different type of manifestation. I was saying at the beginning that we are multisystemic. So we have gastrointestinal, we have development, we have brain, we have vascular, we have respiratory. So we are going to choose not only one aspect, but we are going to choose different aspects, so that what we call domains, and we are going to group them together so that it can reflect the overall disease impact. It doesn't mean that we are going to measure everything, but we are going to select the domain in relevance of the maximum of patients that we can include in those domains, and in if there are way to measures that are standardized and relatively easy and already tested in other diseases. So we have to juggle between the juggle, sorry, not juggle, between the type of domain that are relevant to the patients and standardized and qualified measurement by the FDA. So instead of focusing on a single endpoint, yeah, like in classical clinical trial, we are going to combine multiple domains into a single composite outcome. And I will talk about which one we chose already. For the acute treatments, we want to focus on one aspect of the disease. So for now, what we are focusing on is tracheal stenosis because we have several patients that have an urgency in this treatment. We want to evaluate the treatment efficacy on a single symptom. And you want to focus on what we call the primary endpoint. So you are going to measure one thing and you are going to base the decision of efficacy, not efficacy, on that primary endpoint. So one endpoint. You might have a secondary endpoint or several secondary endpoints that are measured more to give an information about the treatments than to see if it's going to be accepted or not. If you have good results on the secondary endpoint, that can benefit also in the decision from the FDA. but the important one is the primary endpoint. If you have good results with secondary endpoints, but your primary endpoint is not good, it will be rejected by the FDA or the EMA. The primary endpoint is the point. So for a clinical trial in Myhre, with all the clinicians, I mean, not all clinicians in the world, but the main clinicians that we have around us, we have worked already on the domain for the chronic treatments. And the 4 domains that we have chosen is skin and joint, cardiac respiratory, gastrointestinal, and behavioral and cognitive. So those are the 4 domains that we are going to study, and we are going to make a composite responder criteria. So by having some responder index for each domain, and then you score each domain and you make a kind of equilibrium between the different domains. Because what we have is that skin, joints, and behavioral and cognitive is in the large majority of patients. Cardiac and respiratory is very important, but in a small proportion of the patient. And gastrointestinal is in between the 2 others. We have a majority of patients with gastrointestinal issues, but it's not as dramatic as the cardiac and respiratory consequences. So those are the 4 points, 4 domains where we want to work. For skin and joint, or COA, that's clinic outcome assessment. Sorry, I should have put that in clear. So for the outcome assessment, so the way we are going to measure the changes for the skin and joint is very standardized. It's the same tool that can be used anywhere in all the different clinics in the world. So we chose the Dermalab and the Dermalab single for elasticity and collagen and dermal thickness that we are going to measure, and the joint with the goniometry to measure the joint range of motion, so how much you can move an articulation. And we might add the ultrasounds as a backup, but the main 2 measurements that are really standardized, agreed by the FDA and the EMA, are the Dermalab and the ROM. So that's for sure. For cardiac and respiratory, it's still under review because there is so much heterogeneity in cardiac and respiratory issues in patients that it's difficult, and we have it's difficult to choose what to measure and standardize it across all patients. And in addition, we have the pediatric versus adults. So measuring respiratory in pediatric is way more difficult than in adults. And we have at the same time a behavioral component of the disease that makes the measurements more difficult. For example, for cardiac and respiratory, everybody thought, well, the 6-minute walk is going to be the thing, because you can see for the respiration and the ability of the cardiac ability, the joint stiffness. So that's great. The 6 minutes walk will work perfectly to summarize Myhre syndrome patients. And then we realized that with a pediatric patient that have ADHD or autism, make that kids walk 6 minutes with being really focused on what they do is going to be a real challenge. So we are looking at other wearables that could take into consideration the fact that the measurement has to be done with behavioral ADHD, autism patients. So that's complicated. So this part is not done. For the gastrointestinal, so the 3rd domain, it's It's mainly surveys that we give to the patients with the PACSIM or the Promise GI. So those are surveys that are qualified and approved by the FDA and the EMA. And it's basically surveys on the spontaneous bowel movements per week. So that's something you measure, and other aspects of gastrointestinal in the Promise GI. For the behavioral and cognitive, we are going to measure, get data on sleep, because that's a main issue. And we have selected different surveys also in Promise. So Promise is where you can find surveys that are agreed that are approved and qualified by the FDA and the EMA, by all the authorities. So that are surveys that are really, if you use them to collect data, you won't have any problem. You don't have to prove that this survey is relevant. It's a survey that is already ready and proven and standardized. So for the cognitive and behavioral, we work with patients already, and we selected 8 different surveys on different aspects of cognition and behavioral. And we are still looking for a PI, so principal investigator, to run the study. But what we want to measure is already there. So we're quite advanced, I would say, on the domains and the COAs. The big one is cardiac and respiratory, where people are still working on it. And the foundation is working on it mainly. Do you have any questions first here? Or remarks, or do you agree with these domains, or do you think there's a domain that we forgot as patients?
But I understand. I agree.
Okay. Anyone else? Anyone has suggestion where we could adapt that? Because that's the patient voice is really important in the domains, because we really want to measure something that is important to you. Okay.
We are not enough people here to.
Yeah. I know.
Yeah.
So going ahead with what's going to happen. There are different phases when we are ready, when we have the treatment, preclinical data that are show really that we have a good chance that could work in humans. Then what do we do? So we have collected all the data that I mentioned before, everything is checked. Then we go to the FDA, and they say, okay, you have a good protocol, you have the proof that it can work, that it would probably work, we don't know. Everything is ready, what do we do? They say, okay, go for it. The first thing to do is recruiting patients. So patients are asked if they would like to participate, and we check for their qualification in regards of inclusion-exclusion criteria. For example, if it's only for pediatric, we are not going to take a patient of 35 years old. So this phase, believe it or not, is the phase where 30% of the rare disease fail. Clinical trial fail because they cannot have enough people. They cannot recruit enough people because the patients do not believe that they should participate in a clinical trial. Either they don't understand, they are not interested. 30%, 1/3 fails there. To me, it's crazy that we cannot have enough patients to save patients. So that's very important. I think it's very important to inform patients to understand that you understand what's a clinical trial, that you understand the risk, that you understand the benefits, and that you make your decision not only based on what you heard on the social media or what you heard from your neighbor, but really take time to analyze the situation and take your decision. So when we have hopefully enough patients, you have in rare disease, we usually cluster phase 1 and 2 at the same time because we don't have a lot of patients. So we do the toxicity study and the side effect study and the efficacy at the same time. So it's phase 1, 2 together, and it's usually between 5 and max 20 patients for rare disease. So we are going to look at the dose, the regimen. We are going to adjust according to adaptive protocol. So in essence, that's where you have one of the different type of protocol. So we take some part of each that we like and we create our own protocol. But in the phase 1 and 2, we have some adaptive that's logical to every rare disease. The duration is usually between 18 and 24 months. So that's long, but we need to, that's for chronic. We need to really see, and usually you include a 6 months of baseline data collection. So you look at you really measure everything, all the domains during 6 months without the treatments, because we don't want to have a placebo group. We want to have everybody on the treatment. So you include 6 months where you are going to measure everything on each patient, and the patient is going to be its self-control. So that will the first 6 months will be the control compared to the 18 months of treatment after that. If everything is okay, we have limited side effects, limited toxicity. I don't say zero side effect and toxicity because it doesn't exist. But limited side effect, limited toxicity, a good efficiency of the treatment. Then we go to the phase 3. So we take other patients, a larger group, bigger than 20 patients. And sometimes with different sites, well, usually, not sometimes, usually on different sites. So you can have 2 or 3 sites in the US, 2 or 3 sites in Europe, measuring exactly the same things in the same conditions, everything equal, and then you can have a larger population. You do again the same duration, 18 to 24 months, and then you hope that it's going to be approved, that you have efficiency, safety, and that you'll get a new treatment. So I think, yeah, that's it. So I'm going to stop sharing, and I would like to have some questions for everyone. So that's the basic of clinical trial. I didn't go in details in every phase, but I would be happy to have your reactions after 45 minutes of me talking, maybe too fast, I don't know. So do you have any questions about
Yes.
what's going to happen? Yeah.
Okay. I have questions kind of going back to last month. Can you give me an example of the kind of things characteristics you'd be looking for in the cell tests? And the secondary is there the cells does that present as a much problem with heterogeneity as the clinical tests?
So the test we have at the cellular level is, maybe I can share that. Where is it?
I mean, I don't have a clear idea of what kind of things you'd be measuring, whether it's chemical or metabolic, or as there structural changes in the cell. I just don't know that.
Yeah, I'm going to show you that one. Can you?
Yeah.
So that's the preclinical platform that we are developing and that we have at 80% of it. So we have the patient-derived iPSCs, so stem cells coming from their blood. And we took the 2 main mutations, the I500V and the R496C. So those are the two mutations from patients. So they are really patient-derived cells. And then we reprogram them in two aspects that are, according to the patient, the most important and the priorities, which is fibrosis and stenosis. And the other one is neurodevelopment. So those two aspects are going to be tested, the treatments are going to be tested on those two aspects, on brain microvascular endothelial cells. So that's the cells that are important in the vessels of your brain, but also representing the vessels everywhere. And those are easy to measure. The fibrosis is easy to measure. I mean, easy, nothing is easy, but that's a good model to measure fibrosis, let's say. So we are measuring fibrosis, wound healing, cells migration, scratch wound test. So we take the cells and you just scratch the cells and you see how they repair, if you want, how
Oh,
they
okay. Okay.
proliferate and go from one side to another. And we measure what we call the end empty. So now we are going really in details. It's endothelial mesenchymal transition. So that's a mechanism that is fundamental for fibrosis, and we measure that mechanism in the patient cells. For the neurodevelopment, we chose the microglial cells because we could choose also, I chose also the astrocytes, but we don't have an infinite amount of money, so we had to choose the most relevant cell type we wanted. We chose the microglia, which are cells that help. I could compare microglial to the immune system if you want in the rest of the body. That would be the immune system in the brain, and that would be the cells that's clean up, if you want. Together with the astrocytes, they are going to clean up and react when they see that there is something wrong. They are going to react and do what we call phagocytosis, which is eating the trash, if you want, and remove the trash. It's more complicated than that, but let's say phagocytosis is really going to eat the things that are bad for you. And at the same time, the microglia are going to guide also the neurons, such as the astrocytes also do this kind of work. And I hope that we'll have enough money one day to develop the astrocytes, because the astrocytes also stabilize the synapse, so the junction between two neurons. They are around that, so that's also important. But for now, what we measure is the microglial reactivity in the brain and all the different aspects of fibrosis, not all of them, but some different aspects of fibrosis in endothelial cells. And then we'll have the patient-derived organoids. So we are working on that, but we don't have them yet. The blood vessel organoid is
And now you're going, I was just mainly interested in the cellular level. I couldn't really
okay. So
visualize what the functions of the cell. I guess that's what I'm ignorant of. So thank you. I got a better appreciation now.
and this picture is really a picture from our patient cells.
Hmm.
And we have also the mice models, humanized mice model. We have two I500V that are one in Harvard, one in the Institute Imagine in Paris. But we as a foundation are developing the R496C mice with a very special design so that we can test directly in vivo the gene editing in these mice.
Okay. Yeah. Thank you.
For the